All right, we're going to go over section 2. Before we go over section 2, let's pose a couple questions for section 1. One, what is a phospholipid? And describe how it's made up. And 2, what are the 4 proteins that are found in the plasma membrane or the cell membrane? And what are their functions? Pause the video right now and go through those questions really quick in your groups. Section 2 is cell transport. So we talk about cell transport. We have 2 types. We have passive and active. Today we're going to talk about passive transport. Passive transport is when we have a movement of materials across the cell membrane without the use of ATP. Now, they will move until they hit an equilibrium state, which means we have the same concentration mixed throughout. So like we take air freshener right now and spray it in the front of the room, you would slowly see it diffuse throughout the entire room until the people in the back of the room could smell it just as well as the people in the front of the room. That's diffusion. So basically all the particles are hitting one another until we can move them all the way throughout the entire classroom. So we move from a high concentration to a low concentration. This is called the concentration gradient. Really important to know for this because this is how we get the movement of particles all the way across the cell membrane. Passive transport is broken into 2 basic types: simple diffusion and facilitated diffusion. In simple diffusion, we don't require ATP because we are passive. We're moving from a high concentration outside the cell membrane to a low concentration inside the cell membrane. However, with this type of transport, we will only see small nonpolar molecules moving because what they're doing is they're actually moving through the plasma membrane. And so things like natural steroids, which are lipids, pass through that cell membrane. We can also see carbon dioxide and oxygen diffuse the same way. With carbon dioxide, we'll see a higher concentration inside the cell than outside the cell. That's because it's a product of cellular respiration. And so after we've used glucose in the cell, carbon dioxide is released out. And so basically carbon dioxide is going to move from the inside the cell to the outside the cell through simple diffusion. Oxygen, which we take in through respiration, is going to be in a higher concentration outside the cell than inside the cell. And so then oxygen will move into the cell through simple diffusion. Again, they don't need any proteins to do this. They can just do it. The second type of diffusion is facilitated diffusion. Facilitated diffusion, again, like simple, does not require ATP, but it has to have a protein. One of our integral proteins, which is all the way through a transmembrane protein. And so that is necessary for facilitated diffusion to occur. And there are 2 types of proteins. One is a channel protein and one is a carrier protein. With facilitated diffusion, it can move large nonpolar molecules, which are just too big to diffuse completely through, like our steroids, carbon dioxide, and oxygen found in simple diffusion. They're just much, much bigger. They cannot go through on their own. And the other one is a polar molecule. Remember we said last lesson that polar molecules cannot move through the plasma membrane without the assistance of a protein. Basically, the nonpolar heads won't let them pass through. And so because they're not able to diffuse easily, we have to use these proteins to go through. So let's talk about channel proteins first. Channel proteins are going to move things like ions, sugars, amino acids, water. These are using channel proteins. Basically, we're seeing right here, this is a channel protein. It's open all the time. And so when it's open all the time, you can see that molecules will move from a high concentration to a low concentration. Now, it could be a high concentration outside the cell moving into the cell, or it could be a high concentration inside the cell moving out of the cell. But they're able to move freely and not have to worry about any closing of this membrane. Now, this can be a good thing or a bad thing. The problem with this membrane or pore being open all the time for things to move into and out of freely based on the concentration gradient can be an issue, because if we have too many or too much of something, it can actually kill our cell. We can't stop it. So your cells will actually explode if there's too much water available. You can't do anything about it. The other one is a carrier protein. A carrier protein has what we call a binding site. You can see that binding site right here where it's actually attaching. And so that is a binding site. The binding site is important because it actually, what we can see right here with our carrier protein, it's closed. It's not letting anything into the cell. And so until it has it attached to the binding site, and so it can regulate this one a little bit. It will bind to it, the cell then opens up, and then it releases the particle inside of the cell. So that part's kind of cool. The problem with this is if you don't have these carrier proteins, so you have some deficiency, it won't let molecules pass into and out of the cell, and that's usually because of like some genetic problem. This is really great though because our cells don't get overloaded with this, or we can create a concentration gradient too. Now, if we're trying to go against a concentration gradient, we're going to have to use proteins, but that's going to be active transport, and that's our next lecture. But for this, we use these carrier proteins. They fit in the binding site. They do not require energy to occur, so they'll actually do that on their own. One of the most important types of facilitated diffusion, which is so important for our body, and you've heard of this probably before, is osmosis. Osmosis is the movement of water into and out of the cell. They will use what we call a water channel. So we have a channel protein, which is open all the time and allows for these water molecules to pass into and out of the cell. Now, water is so important because a lot of materials will dissolve in water and can be easily transported into and out of the cell by the water itself. Now, our blood and the water in our bodies is not pure distilled water. It's really a lot of saline solution, which means it has just a very, very small amount of salt particles mixed into it. And so we always have this like slight salty blood or inside the cell. So because of that, though, we're able to push water into cells that are dehydrated and push water out of cells when we are dehydrated. And so water concentration becomes really, really important. However, do understand when we do move water, it's from a high concentration of water to a low concentration of water. So if you have a cell that is, say, you just decided you're not going to drink water for 2 days and all you drink was Coke. Well, there is water in Coke, don't get me wrong. However, there's a lot of other stuff in Coke too. If before this time you just drink water all the time, and decided to go on a Coke spree for 2 days and no water. Basically what's going to happen is your blood is now going to have a low concentration of water and a high concentration of sugar and salts and all these other preservatives in it from the Coke you've been drinking. And so your cells, though, because you were drinking a lot of water before the Coke, have high concentrations of water in them. So basically what you're going to see happen is your body is going to try to equal out or create an equilibrium of these water molecules. And so we are going to see the water move out of your cells into your blood to try to increase the amount of water in your blood. And basically it's diluting all the other junk that you were taking in by drinking just strictly Coke. It'll do this until you reach an equilibrium state. This is also why when you take meat and you throw salt on it, what happens to the meat? Well, it preserves it. It's actually removing all the water from the meat, and it's dehydrating it out, so it will preserve longer. They do the same thing with mummies when they the same thing with mummies when they started to mummification process on there. Some of you may remember the chicken leg with Mr. Gunn. In 6th grade, same concept. They're actually putting all that salt in there, and it's dehydrating, it's taking all the water out of the cells, putting it into the salt solution, dehydrating that. And so osmosis is simply a type of facilitated diffusion. You don't need any energy for that. Now, the movement of water is done in 3 ways. We have these big sciencey terms for it. They're called isotonic, hypotonic, and hypertonic. So let's talk about the solution first. So if I have an isotonic solution, which we see right here, it means that there's an equilibrium that's been reached. Isotonic means we basically have no net movement of water. You have water still moving into the cell, but the same amount is moving out of the cell. So the cell and the solution itself are both isotonic. And so we don't see the cell getting any bigger or any smaller. In this example, we have red blood cells. In a hypertonic solution, which is right here, the hypertonic solution has a high concentration of solute. So you can see all the little particles around. Those are going to be solute particles. And so the solute particles, since they are such a high concentration, it tells us that we don't have that much water in that hypertonic solution. And so the cell actually had more water. It's like you drinking the Coke. And so all the water is going to move out of the cell, trying to dilute these particles here. When we have this type, we have a hypertonic solution. The cell is actually hypotonic to the solution, which means all the water is going to move out of the cell. In the hypotonic solution, we don't have a whole lot of salt particles here. That means there's a lot of water, high amount of water, low amount of solute. And so what's going to happen here is we are going to see water move into the cell because we have a high concentration of water and it's moving to a low concentration, which would be inside the cell. Now, in the hypotonic solution, the cell would be hypertonic to that. And so basically we're going to see water move from a hypotonic to a hypertonic. So when we talk about the cells and all the water and what's going on in them, the cell will actually be hypertonic to a hypotonic situation. So, and then in this case, we have a hypotonic solution. The cell is hypertonic. So we have to go from a hypotonic state to a hypertonic state, and we're trying to reach an isotonic or a normal. Now, this is in animal cells. We like to be isotonic. We want to have the same amount of solute into and out of the cell. We want to see a net movement that's absolutely the same. And we like to be in a isotonic state. However, plants aren't that way. Plant cells are typically hypertonic, and they like to have an hypotonic solution on the outside, which means water is constantly going into the cell. So again, plant cells like to be hypertonic to a hypotonic solution, which means water is constantly going in. This allows them to stand up straight. And so if you look at the plants around the room, if they're wilting, it means their cells were hypotonic to a hypertonic solution, and the water moved out of the cell. Plant cells have a large central vacuole which holds water. It allows them to have turgor pressure, which is really important for them to maintain this isotonic state. Okay, so tomorrow you are going to do a lab called the Gummy Bear Lab, and you're going to kind of recreate some of these scenarios. So you're going to watch a video. In that video, then you have to talk about your isotonic, hypotonic, and hypertonic states, and you are going to then recreate that video. Before we get done for today, I do have a second video that you're going to watch talking about just simply water movement. And so remember, you have a homework assignment after this. It's a worksheet, and make sure that you do pay attention to the video tomorrow because it's going to be important to get all that information down. All right, have a great day.